Howdy, this is Mr. Justice, and in this video we're going to talk about the types of mutations, and we'll be filling out these guided notes. Now, you can make a copy of these guided notes by going to crjust.us/MUT if you'd like to quickly make a copy of these notes right here. You can push pause on this video to do that. All right, so let's go through and talk about what mutations are. So a mutation is a change in a DNA sequence. I'd like to actually highlight that because that sounds pretty important. So I'm going to select change in a DNA sequence, and I'll come up here and use the highlight tool. And that is right up here. It says highlight color up in my toolbar. Click that and choose a nice color that's easy to see. All right, so mutations can result from DNA copying mistakes made during cell division, exposure to ionizing radiation, exposure to chemicals called mutagens, or even infection by viruses. These germline mutations can occur in the eggs and cells and can be passed on to offspring, while somatic mutations occur in body cells that are not passed on. So to complete A here, what I'd like us to do is, you can push pause, but go ahead and explore a couple of the terms up here. In this paragraph, for example, ionizing radiation, what you could do is highlight ionizing radiation and then right-click on it with your mouse, and then come over here and click on Explore. And when you do that, information about ionizing radiation, even images associated with it, will pop up. Let's take a look. Ionizing radiation refers to the particles and the high-energy light that are emitted from the nuclei of radioactive elements. Alpha particles can be emitted from a very large radioactive element in an effort for that element to, or atom rather, to expel particles, protons, neutrons, to stabilize itself. Now, alpha particles that are emitted from radioactive atoms have energy, have velocity to them, but not enough to pierce through your skin. The problem with alpha particles is that if you breathe them in, these alpha particles can get into and destroy the integrity of our cells that make up our lungs. Same thing with beta particles, also particles that are emitted from a radioactive unstable nucleus of a radioactive atom. The ones that are really scary are the gamma ray radiation, X-ray radiation. These types of ionizing radiation here are high energy light. Think even higher energy than UV light. X-rays and gamma rays have enough energy to pierce through your clothing, your skin, and break up the DNA found inside of your cells. So if there's like gamma ray radiation or a radioactive element that is actively producing any of these particles, it would be best to get away from it or get behind a concrete wall or a leaded wall. In fact, one of the reasons why your doctors will cover up your body with a leaded apron is to prevent the X-rays from mutating your cells in your body. So that's the type of ionizing radiation, and I've selected it. Go ahead and highlight that too. DNA copying mistakes, that's going to be done by DNA polymerase. Don't forget, DNA polymerase is this enzyme that makes a copy of your DNA, right? Think about as we grow from one cell to 2 and 2 to 4 and 4 to 8 and on, we've got to make sure that we have an exact replica of our DNA in each of those new cells. So DNA polymerase it will make a mistake 1 out of every billion nucleotides or so. Remember that DNA polymerase has proofreading capabilities too, to ensure that those mutations don't occur, but every so often they do. So that's another source of mutations. Chemicals, like the chemicals in cigarettes, or the chemicals used in a chemical called asbestos. Asbestos is used in insulating walls of homes and in offices earlier in the 20th century. The problem, and it does a really good job at that, the problem with asbestos and these chemicals is that, it can break off in very small sizes. This particulate matter can enter into your lungs and lodge themselves within the cells, the alveoli that make up your your lungs and causing problems. It's a mutagen, so it can create mutations in your DNA, which can have profound effects on the ultimate proteins that are produced. And also viruses, too. Scientists believe that about 8% of the human genome is actually made up of viral DNA. These viruses, like retroviruses, infect a cell. These are our ancestors, right? Our common ancestors millions of years ago. These retroviruses infect the cell, and then there's machinery inside of those viruses that allow that DNA to be integrated into the human genome. And I want to point out here that mutations aren't always bad. In fact, these mutations from viruses and DNA copying mistakes can really drive evolution. These changes in the traits may give an advantage to the organism that has that mutation in it. And if the advantage allows it to live to its highest biotic potential, then that trait will likely be passed on to their offspring, their progeny. But mutations can also have disastrous effects too. They can also lead to diseases, cancer, and other issues. So take a look here. What I'd like to in A do is create a list. It has a little bit more information in it, and if you'd like to, you can just copy what I have here on this screen. Let me make this a different color so that you can see it. DNA polymerase makes errors. High energy light, UV, gamma, X-ray has enough energy to cause mutations. Chemicals, and I've given you two examples of cigarettes and asbestos. And then those retroviruses insert viral DNA into the human genome. So earlier in this paragraph, it says germline mutations occur in the egg and sperm. So germline cells are the cells that give rise to or differentiate into the reproductive cells. These are called gametes, or the egg and the sperm here. And if a mutation were to occur to the cells that ultimately differentiate into eggs and sperm, and those eggs, sperm are fertilized, then that mutation can be passed on to the offspring. So what are the implications then if a mutation were to occur? Well, take a look. The mutation to the DNA in the germline cells means the mutation can be passed on to the progeny, the offspring children, causing an increase in the likelihood of childhood disease, cancer, or birth defects. Mutations to somatic cells, those are everything other than the reproductive cells. Think of like your skin, for example, right? Mutations that you might get from the UV sun, from the UV light that comes from the sun, might scramble up, mutate, destroy our DNA in the cells in our skin. But since these cells, our somatic cells, are not passed on to offspring, those mutations will stay with the organism. All right, take a look here at what Dr. Francis Collins says about mutations. I'll read it to you, follow along on the screen. He said, mutations have been the source of many Hollywood movies, but it's a really simple process of a mistake made in a DNA sequence as it's being copied. Some of that's just the background noise that DNA copying is not perfect, and we should be glad of that, or evolution couldn't operate. But mutations can be induced by things like radiation or carcinogens in a way that can increase the risk of cancers or birth defects. But it's pretty simple. It's basically an induced misspelling of the DNA sequence. That's a mutation. Take a look at C here. Dr. Collins says that a mutation is an induced misspelling of the DNA sequence. Use the language of DNA, create a 9 DNA nucleotide sequence before and after a mutation. For the mutated DNA sequence, either add or delete one nucleotide or substitute one nucleotide for another. So this is what it might look like if you were to do that. If you like, you can just use mine. What I'm going to do is write out the word before. I'll go with A, T, 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 G, G, G. Keep it simple. And so if a mutation were to occur, think about this for a moment, right? After the fact. So if we add in or delete one of the nucleotides, think about what will happen then to this sequence of 9 nucleotides, right? Think about it. If I remove this A, these nucleotides are going to regroup. And all of a sudden now, this deletion event has resulted in really 8 new nucleotides that weren't there before, a new order of those nucleotides, right? Hey, let's see what an addition might do. Let's say that a guanine, is added at that second position. Well, I'll go ahead and shift all of these nucleotides over one to the right, and all of a sudden, again, we've got a completely different sequence of nucleotides here. What are the implications for the resulting protein? Well, we'll see what those have soon. Real quick, I'll show you what the other ones might look like. Substitution would just be this, right? Imagine that this T was substituted for, like, let's say a cytosine. That could happen, sure. So choose one of those types. I think I'm going to go with, I'm going to go with this one. Adding a nucleotide in the middle and shifting the other nucleotides over one to the right. All right, and we'll come back to this at the end of our lecture here. So let's keep going, scroll all the way down. All right, so what we're going to do here is annotate this little photo here that's got the 5 different types of mutations here that we're going to learn about today. And so in order to be able to annotate this, what I'd like to do is double click on this image. And what it will do is pull up a Google drawing with all of the tools at the top that you can use to help yourself annotate. What I'd like to do, actually, I'm going to use this magnifying glass here and zoom in a little bit so that we can see a little bit clearer. All right, so the first type of mutation here is called a point mutation. There are 3 different types of point mutations: a silent, a missense, and a nonsense point mutation. So let's do this. I'm going to grab the line tool up here in the toolbar and draw a line underneath. You know, I'd like to actually increase the size of this line, so I'm going to come up here to the line weight, and let's change it to 3 pixels. And I'll redraw this or change the color from black to green so it really pops right there. Kind of cool. So a silent point mutation has no effect on the protein sequence at all. Take a look at the before sequence on the left and compare it to the after sequence on the right. Take a look. The DNA has been, this A here has been substituted for a thymine. Take a look. We've got a new mRNA sequence over here, but look at the sequence of the amino acids once we translate that mRNA. Serine, valine, proline, tyrosine. It's the same sequence as serine, valine, proline, tyrosine on the mutated side over here. The silent mutation, point mutation, has no effect on the protein sequence. It's the only one of these mutations that don't give rise to new traits, new expressions of these genes. Why? Because his valine in the second position is also here after the mutation in the second position. So there's no net change to the protein. Let's go ahead and change up the shape. I'll grab the square. And what I'd like to do is draw a box around the differences here on the right-hand side. And take a look. When I let go of that box right there, I can't see the differences. Take a look. Let's edit just a little bit. Let's come up here to the fill color. It looks like the paint can. Click on that. Click transparent. And I'd like to make everything match. And so I'll grab that 3 pixel edition of our rectangle here. And then let's change the color of that rectangle to match. Green. All right, the next type of point mutation here is a missense point mutation. Let me grab my line again. So I can underline. By the way, if you'd like to make your line nice and straight just like this, as you pull that line from left to right, hold your shift button down on your keyboard and then let it go. And you'll get that nice, perfectly horizontal line. Let's go ahead and change the size and the color to match. A missense point mutation results in an amino acid substitution. Take a look. After the mutation, which happens to right here, looks like this first C. it has been substituted for an A. Well, what effect did that have on the amino acids? Look, that third position is a proline. After the mutation, that third position is a threonine. Different. So that right there, this missense point mutation results in an amino acid substitution. We've substituted a proline for a threonine. Let's go ahead and grab our shape tool, and we'll grab and draw a line around this. And make it transparent, change the color, make it 3 pixels. A nonsense point mutation here is going to substitute a stop codon for an amino acid. So take a look at this, right? Well, that last C has been substituted for a G. What effect did that have on the overall protein sequence? Serine, valine, proline, tyrosine. Take a look. That tyrosine has been converted to a stop codon. So let's go ahead and draw that line underneath nonsense right here. One way that you can remember nonsense point mutations is by remembering the word non-stop. Non-stop. Nonsense, stop codon. Let's go ahead and highlight the difference here. Oh my gosh, I'm trying to grab there we go, the shape. Doing it wrong. There we go. Let's highlight this. And I'm going to add, see this little text box here? Pull out the text box rather, and let's type in non-stop to help us remember. I'm going to make this a little bit smaller so that it fits my very small area here. There we go. Bold it. Maybe make it a little bit bigger. And what if we made this like red? Just changing the colors, making it our own. All right. Maybe put a little quote. Non-stop. Or nonsense point mutation makes a stop codon. All right, the other type of mutations here are called frame shift mutations, and there's 2 types of those, either an insertion or deletion frame shift mutation. Let's go ahead and draw a line under this so it pops. Now, take a look, right? It's best to just look at the example here. This T and A, the thymine and the adenine on the DNA on the left side has been removed. And so because of that, think about this, the reading frame has shifted 2 over to the right. So now we have all new codons here, right? GUA is now GCC. CCC is now CUA. And UAC is now CUU. There's a massive amount of difference. These frame shift mutations are going to result in very, very different amino acid sequences that very likely won't work. You've got to remember about these proteins here. Right? We see proteins, but they're made up of amino acids. And each of these amino acids has very specific chemical properties that allow this sequence of amino acids to fold into a specific shape. When we deal with proteins, form determines function. So if the protein folds into its correct form, its function will be there. If it doesn't, let's say there was some DNA that was deleted and it resulted in a completely new string of amino acids, that protein might not fold into its correct shape. And if that happens, form determines function, it won't work. It doesn't have the form, it won't have the function, whatever that function of that protein is. So what I'd like to do is highlight these differences. I'll grab the shape tool, and since this is a disastrous mutation here, I'm going to highlight all of those differences. All of the differences, right? All 3 of those amino acids have changed. Let's go ahead and format this a little bit. And there we go. So once you're happy with your notes here that we've annotated, go ahead and click Save and Close. And what you'll find here is, boom, we've got some notes. So what I'd like to do here with our last part is go back to our before and our after. And what I'd like to do is look to see what effect that mutation had. So I just copied and pasted my before and after. Now, there's a lot of thymine here, so we've got to think about this. This is mRNA, or DNA that we've got to convert to mRNA. So AAA is going to be UUU, TTT is going to be AAA, and GGG will be CCC. After my mutation here, right? Think about, let's type out our mRNA. It's going to be UGU. ATT will be UAA. TGG will be ACC. And this one will be AC. All right, so there you go. Take a look. Let's now translate these. So now we're going to make here, let me just do this so I have a little bit more room. There we go. Okay, so let's make this into that sequence of amino acids. So if I look up UUU on my universal genetic code here, let's use the one from this worksheet. Let's see, UUU is phenylalanine, so let's come back. Phenylalanine, we'll put a little dash in there. The next codon here is AAA. Let's go to lysine. And CCC, proline. All right, so this is the before, right? Remember, there's no mutation here whatsoever. So let's see what happens then when we inserted that extra nucleotide into the sequence here. We've changed the mRNA, we already know that. Let's look to see what effect that has on the amino acids. So UGU is cysteine. UAA, stop. Look at that. Stop. So that mutation right there, this insertion mutation has resulted in a stop codon. Not good. Disastrous effects on the resulting amino acid. It should have been phenolphthalein, lysine, proline. Instead, it's just cysteine. That's not an amino acid that looks anything like the one before. And so this can have some big consequences. I hope that made sense. Let me know if you have any questions.